Hi dear students, a warm good morning to all of you and welcome back to one more session of Microbial Taxonomy. The previous class you came across the different concepts of taxonomy, like it mainly consists of classification, nomenclature as well as the identification. And we also came across the different types of classification systems and what are the levels of different classifications or the taxons which we use in taxonomy. Now, today we're going to come across what are the techniques we used for determining microbial taxonomy and phylogeny. The first and foremost is the classical characteristics. The classical characteristics are the ones which were used from long back itself. It's not that we don't use it right now, but we also do consider them. And the classical characteristics includes the morphological characteristics of a bacteria, the physiological factors, the biochemical factors, the ecological factors, and the genetic factors of a bacteria. Now, what do you mean by the morphology? The morphology, in fact, consists of different other parameters. Like it can be what are the different shapes of it, and how is it, like how does it have a flagella, does it have a cilia, like that. These all come under morphological characteristics. Now coming to the physiological characteristics, you can have different types. Now some bacteria can utilize high NaCl concentration. Some of them could live in alkaline conditions. Some of them can live in acidic environments. Some can utilize specialized sugars, some cannot. These are all coming under the physicochemical reactions or the physicochemical characteristics, that is based upon their physiology. Now, aerobic bacteria are there, anaerobic bacteria are there. These are so the physiology of the bacteria as well as its biochemical characteristics will also contribute to the classical characteristics. So physiological could include aerobic, anaerobic, then there would be some will be strict anaerobes and like that. There are lots of characteristics. Then the ecological characteristics, how it lives, where it lives, that is it. And its environment, how it is. Then the genetic characteristics, does it have a plasmid, about its genetic elements associated with this. All these characteristics come into the classical characteristics. So we'll go into the morphological characteristics. As I told you, the cell shape, the cell size, the colony morphology. The colony morphology means when you grow it on a agar plate, how does the bacterial colony look like? You have got lots of subdivisions in it, like you have colony color, the colony shape, how the colony is raised, or it is flat, or is it convex. These are all coming under colony morphology. Or is it swarming type of colony, that is moving, like some colonies will try to move across a plate. Move means when they grow they will have a literal movement. And the ultrastructural characteristics. Ultrastructure means inside the cell, how is it? The staining behavior of the bacteria, whether it has cilia and flagella, how does it move? Like does it have a gliding back motility or a spiral movement, or what is the endospore shape and the location? Endospore is a spore which has been formed within the bacteria. So how these endospores could be either be central or sometimes it might be at one end and all that. So based upon that, the endospore shape as well as the location, you can classify the bacteria. The spore morphology, is it how is the spore look like? The cellular inclusions. The cellular inclusions mainly includes different types of within the cell, like you have magnetosomes, phbs and all that. So based upon the cellular inclusions, the colony colors, these all come under the morphological characteristics. Then the physiological and the metabolical characteristics, as I told you, the sometimes the oxygen variation, that could be of characteristics. Then the carbon and nitrogen sources, the ones which it can use. Some bacteria can use some carbon sources, some can use other, the cell wall constituents, the energy sources, the fermentation products, the general nutritional type. the temperature optimum and its range. And some bacteria also do have the property of luminescence. Luminescence means to glow or to produce light. The mechanism of energy conversion, osmotic tolerance. Osmotic tolerance means the tolerance to salt concentrations. The variations in salt concentrations will cause osmotic imbalances. Then the pH optimum, the growth range, the photosynthetic pigments, the salt requirements and tolerance, the secondary metabolites formed. Secondary metabolites are something which the bacteria forms when it is in the lag phase of the growth, like antibiotics. Some of antibiotics as well as various other metabolites are also there. These are called the secondary metabolites. They are not needed by the bacteria for its growth, but the bacteria produces it as an additional factor when it is growing. Then the sensitivity to metabolic inhibitors, antibiotics, the storage inclusions, these are all characteristics which will help us to classify the bacteria. The ecological characteristics, we have the life cycle patterns, the symbiotic relationships which they can do. Symbiosis means without causing any harm if they live together. Then the ability to cause disease, the habitat preferences, that is what I mentioned earlier, the environment in which it grows, then the growth requirements. These are all coming under the ecological characteristics. Now, after that, we have the molecular approaches or the genetic approaches. Now, molecular approaches, they play a very important role in identifying bacteria because right now many of the microbes they don't even give a fossil record because they're too tiny to leave a fossil record. But using the genetic material, you will get more amount of large and accurate data. And using these molecular approaches, we get to collect a large and accurate data from many organisms. And you also can maintain various phylogenetic inferences based upon this molecular data also. Now let's see how does the molecular approaches help you for that. So coming to the molecular characteristics, molecular characterization has been carried out by using the biomolecules. Now, what do you consider the biomolecules? It could be the DNA, it could be the RNA, or it could be even the proteins. Because these are the biomolecules which mainly contribute to the functioning, or it gives it gives an idea about the functioning of the particular cell. Now, most frequently, we use nucleic acids and okay, we also use amino acids also for the characterization. And some of the techniques which we use for the molecular characterization of microorganisms include the nucleic acid based composition, the nucleic acid hybridization, nucleic acid sequencing, the genomic fingerprinting, as well as the amino acid sequencing. These terms, when you see them, you might find them bizarre and very much confusing, or you might find them complex. But once you go into the details of it, it's quite interesting. So now let's talk about the nucleic acid base composition. We all know that DNA is made up of what? Adenine, guanine, cytosine, and as well as thymine, A, T, G, and C. Regardless of what form you have, all the DNA is made up of adenine, thymine, guanine, and cytosine. What are these? These are the the bases which are being found in the nucleic acids. And among these, the G plus C content or the guanine and the cytosine content will help us to show the relatedness between various organisms. And the percentage of G plus C is equal to the amount of G plus C in the bacteria or the organism divided by the whole number of bases, that is G plus C plus A plus T into 100. That is a percentage of G plus C. And you can see that if the G plus C content has been found to be what is it? the variation of that, if it is less than 10 percentage, then we can consider them as a particular genus. So the people in a or the people or the even the microbes, okay, if the G plus C content has been varied by less than 10 percentage, then you can include that group of organisms in a particular genus. Now you would have a thought like how to calculate the G plus C content. If you take a DNA, the G plus C content is also been related to the melting temperature or the Tm. Now just look at this graph. I will just explain to you how do you get the Tm or the melting temperature. If you take double-stranded DNA and heat it at different temperatures, you know now this is 70, 80, 90 and 100. As you heat the DNA, what happens? The double-stranded DNA will get separated into single-stranded DNA. Okay, and the absorbance of the DNA would also be changed. Now, what do you mean by absorbance of DNA? The DNA can absorb light at a specific wavelength. That is, when you put a wavelength, a light of about 260 nanometer, the absorption of that particular light is maximum for DNA. So you can prepare that in a spectrophotometer, UV-based spectrophotometer and all. You can expose DNA to this particular wavelength. So what you do is, after heating the DNA at different temperatures, you will take that separated DNA and check its absorbance at using a UV-based spectrophotometer at 260 nanometer. And you can see that when you plot the absorbance of the DNA at different temperatures. Now I took the DNA, heated it at 70 degrees centigrade and I kept it. I checked the absorbance. I got about 1 point something, little bit above 1, but not 1.1. And as the temperature is increasing, you can see that the absorbance of the DNA is increasing. And further on, it becomes almost the stable. So there is a temperature at which the whole DNA is becoming single-stranded. When the whole DNA becomes single-stranded, that is it, the time at which the absorbance increases. And the melting temperature is mainly defined as, what is it? When you extrapolate, it is the temperature at which half the molecules of these DNA becomes single-stranded. See, that's what. You have a curve here, which is being found to be rising. That, the middle of this curve, if you put it down or if you extrapolate down, you get the Tm or the melting temperature. And using this, so this melting temperature is directly related to the G plus C content of the organism also. Okay, so greater the G plus C content of an organism, the greater is its melting temperature. So if you have a less G plus C content, the melting temperature of that organism will be found to be different. So by using the nucleic acid base composition, that is the G plus C content of the organism, we can find out whether the organisms are being found to be related to each other. And how to calculate the G plus C content? That is mainly by taking the absorbance of the DNA at different temperatures. By increasing temperatures, and you find out the melting temperature at which you will have to extrapolate and draw this graph. And from the graph, you calculate the melting temperature of the DNA. So if you have two different bacteria or two different organisms, you will take its DNA, check for its nucleic acid base composition, and based upon that you can characterize what is the melting temperature as well as the G plus C content. Now you can see there are different genus of bacteria itself. They have different G plus C content. So if the G plus C content has been found to be similar, then you say that they would be of a particular genus. Now, I do not say that it is exclusively the G plus C content that we use to calculate which genus, because we use different characteristics. I have told you it's a polyphasic taxonomy that we follow right now. And G plus C content is one factor which will enable us to classify them, probably determine what particular genus they would be of. Now here you can have Mycobacteria, which is having a G plus C content of 62 to 70. Escherichia is having 48 to 59. Like that, it keeps on varying. So using the G plus C content, you get an idea. If the G plus C content is less than, the variation is less than 10%, then we consider that they are of a particular genus. The second type of molecular method which we have is a nucleic acid hybridization method. Now, what is a nucleic acid hybridization? It is a DNA-DNA hybridization technology. And here we mainly check whether the sequence homology or the sequence similarity based association between the organisms are being used. What you do is you take DNA, okay, and you convert it into a single stranded one by denaturing it, okay, either by heating or by chemicals you can do it. You will convert it into single stranded, and what you do is you bind the non-radioactive DNA to a nitrocellulose filter. So the single stranded DNA which you put is transferred to another membrane called the nitrocellulose filter. And after that, you will incubate this filter paper along with radioactive other single-stranded DNA. So you have your DNA of interest, which is converted into single-stranded one, and it is transferred onto a nitrocellulose filter paper, okay, or nitrocellulose membrane, and that has been placed in a trough which will already contain radioactive single-stranded DNA. And if this radioactive DNA and your non-radioactive DNA is been found to be similar in nature, or if they have some sequence homology, the radioactive DNA will get attached to the filter. And then after that, you will check the amount of radioactivity. Okay, so based upon this, you say that whether the radioactive DNA and the non-radioactive DNA, if they can bind with each other, it indicates that they have some similarity. So based upon that, you can say whether the two organisms or the two DNAs which you have taken are related to each other. This is the depiction of it. So you have your target DNA. Your target DNA is taken and it is being converted into single stranded. It is transferred to a membrane, and this is the transfer membrane. And onto this transfer membrane, what you do is you will put your prop DNA. The prop DNA is now the one which I told in the last case, it was a radioactive one. And this radioactive DNA along with this membrane is placed, and sometimes we would also transfer it to a X-ray film so that we can visualize it. So what happens is where all this radioactive or the prop DNA can go and bind on the X-ray film, you will get locations. So from that you can understand that okay, this DNA, okay, or this radioactive DNA is able to bind on the target DNA at different sites. And this is a principle of nucleic acid hybridization. And let me just show you a practical depiction of that with respect to taxonomy. This is a picture which I got from the Population and Evolutionary Genetics by Leon. And in this, they have not gone into the further details, but they have used the same DNA hybridization and they have depicted their results. They took 3 different types of DNA, the human, fly, as well as the chimp. And of course, they gave a they denatured it and made it unwind. And what happened is they probed it with the different DNA. Now here fly and human DNA were tried to be probed, and here in this case it was the chimp and the human DNA was tried to be probed, and you can they were being allowed to be hybridized, and you can see that the human and the chimp has been found to be having more conservation. So in this case, or in this example, probably the probe which was being used was being considered to be as human DNA. So, and they are checking whether the human DNA is getting probed with the two different target DNAs. One is a fly and the second one is a chimp. And based upon this, we can see that what? The most similarity is there between human and the chimp DNA because they are getting what? Hybridized. But here they are having less conservation. And it was the DNA hybridization technique which they utilized to find out this particular concept. Now, the next one is a nucleic acid sequencing technique. Here, nucleic acid sequencing is I will just explain it in a very simple manner because the more details of sequencing you will be learning in your coming semesters. Now, what is nucleic acid sequencing? It's nothing but decoding how the DNA is being arranged. Now, if you have a name, I can explain it with an example. If my name is Cheryl, if I want to decode it, I can say the letters S, H, A, R, R, E, L. That is, the alphabets should be arranged in this order. Only then it becomes my name. The same way, if I try to decode the DNA, if I say that A, T, G, C, 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 like that, only if you arranged how the bases are being arranged in my DNA, that is referred to as nucleic acid sequencing. And in taxonomic point of view, we use the small subunit ribosomal RNAs to identify bacteria. You can see that when you take the 16S ribosomal RNA of bacteria, that has been found to be highly conserved. That is, the variations among bacteria is mainly been concentrated in the 16S rRNA region. In other regions, they might be found to be similar, but in the 16S rRNA region, they have things which are unique to their own self. So if you take the 16S rRNA region of the bacteria and then sequence it, you can say into which taxonomic level, okay, into which genus level you can include that bacteria. And the 18S ribosomal rRNA is mainly being used for the taxonomic identification of fungi. Like that, you have different organisms have different genes which we sequence to identify them by nucleic acid sequencing. In the case of bacteria, we have the 16S ribosomal RNA. In the case of fungi, we have the 18S ribosomal RNA. Now, if it is a plant, we have the CO1 enzyme, the Rubisco enzyme or the RBCL type. No, for the plants, we use a Rubisco or the RBCL. And for respiring organisms, we use a CO1, the cytochrome oxidase based genes. So, one particular gene which can help us to identify the organism is taken and its DNA sequences are being coded. I guess we can continue with the other molecular methods in the next class. Thank you for now.